In 2020, a team of Chinese researchers published something that quietly reshaped how doctors think about genetics. They had sequenced the full genomes of over 10,000 people across China, cross-referencing the data against the massive genetic databases already built from European populations. The project was called China Map, and the goal sounded almost mundane on paper. Map genetic variation across China's population to improve disease prediction and drug safety. But when the numbers came back, they revealed something that a lot of Western medicine had quietly assumed was not true. The genetic blueprint behind disease risk, drug metabolism, and basic body chemistry was not the same from population to population. Not slightly different. In some cases, dramatically different. And the implications reached into hospitals, pharmacies, and genetics labs around the world almost immediately. This is the story of what happened when scientists finally put Chinese and European DNA side by side at scale, and why the results forced medicine to rethink some of its most basic assumptions. Before we get into it, hit subscribe and turn on notifications, because we publish new genetic science documentaries every week, and this is one of the most consequential comparisons modern genomics has ever produced. Stay until the end, because there's a twist involving a single gene that a huge percentage of the world's population doesn't even realize is quietly rewriting how they process everyday medication. To understand why this shocked researchers, you first need to understand what most people assumed going in. For decades, the mainstream understanding of human genetics followed a fairly comforting narrative. Modern humans evolved in East Africa somewhere around 300,000 years ago. Around 60 to 70,000 years ago, small groups began migrating out of the continent, spreading across Europe, Asia, and eventually the rest of the world. Over tens of thousands of years, these separated populations developed different surface features: skin tone, eye shape, hair texture, body build. But underneath the surface, the genome itself was assumed to be almost interchangeable. Well, sure, scientists already knew about a handful of famous exceptions: lactose tolerance in Northern Europeans, altitude adaptation in Tibetans. But these were treated as minor footnotes, isolated quirks, layered on top of an otherwise nearly identical genetic blueprint shared by everyone on Earth. That assumption held up reasonably well for a long time, mostly because almost all large-scale human genetic research, going all the way back to the original Human Genome Project, was built overwhelmingly from European ancestry samples. It's a detail that sounds small but turned out to matter enormously. Genetic risk scores, drug dosing guidelines, disease prediction models, all of it was calibrated almost entirely on European genomes, and then applied globally as if it would translate cleanly to everyone else. Nobody had really tested that assumption at scale until China's own genomic research infrastructure caught up. That's exactly what projects like China Map, the Taiwan Biobank, and the Hong Kong Genome Project set out to do. Between roughly 2018 and the present, these programs sequenced the genomes of well over 100,000 people of Chinese ancestry combined, some through deep whole genome sequencing, others through dense genotyping arrays covering hundreds of thousands of genetic markers. The purpose was practical and, frankly, overdue. China has over 1.84 billion people, and almost none of the existing precision medicine tools built in the West had ever been properly validated on Chinese genomes. Researchers wanted better disease prediction, safer drug prescribing, and genetic screening that actually reflected the population it was meant to serve. But almost immediately, the comparisons against European genetic databases started throwing up numbers that didn't behave the way anyone expected. The first surprise came from something researchers call population structure. Essentially, how genetically similar or different subgroups within a single population turn out to be. Scientists expected some regional variation within China, given the country's size and history, but the degree of it caught people off guard. Southern Chinese populations showed genetic variation patterns that were meaningfully distinct from northern Chinese populations, shaped by thousands of years of different migration routes, regional isolation, and admixture with neighboring groups across Southeast Asia. In some analyses, the genetic distance between a Han Chinese person from the north and a Han Chinese person from the south rivaled the genetic distance seen between entirely different European countries. That alone told researchers that treating Chinese or even Han Chinese as one single genetic category, the way a lot of earlier medical research had, was an oversimplification with real consequences for how disease risk gets calculated. But the more consequential discovery wasn't about internal variation within China. It was about the specific genetic differences between Chinese and European populations and how those differences showed up in things that actually matter for health. Take a gene called ADAR. It's one of the clearest, best documented examples of a genetic difference between populations that isn't cosmetic trivia. It reflects an entire population undergoing rapid, distinct evolutionary pressure after the split from other human lineages, and it happened fast, over just a few thousand years rather than the hundreds of thousands of years most evolutionary change requires. That kind of rapid population-specific evolution turned out to be far more common than researchers had assumed. Selective sweeps, sudden bursts of a genetic variant spreading through a population because it offers a major advantage, showed up again and again when comparing European and East Asian genomes, just in completely different genes tied to completely different environmental pressures. European populations carry well-documented sweeps tied to skin depigmentation and lactose tolerance, both linked to surviving in low-sunlight northern climates, where vitamin D synthesis and dairy farming became critical for survival. Chinese and other East Asian populations show a very different signature of selective pressure, most strikingly around starch metabolism. The AMY1 gene produces an enzyme called salivary amylase, which breaks down starch before it even reaches the stomach. Populations with a long history of starch-heavy agricultural diets tend to carry more copies of this gene. And East Asian populations, with a rice cultivation history stretching back well over 10,000 years, generally show higher AMY1 copy numbers on average than populations with a more recent agricultural or dairy-based history. More gene copies mean more enzyme, which means more efficient starch breakdown, a direct genetic fingerprint of thousands of years spent living on rice-based diets. These dietary adaptations don't stay isolated in one corner of biology either. Starch metabolism feeds directly into insulin response, which feeds into fat storage patterns, which feeds into long-term cardiovascular and metabolic disease risk. And that's exactly where researchers found some of the most clinically important differences of all. Take type 2 diabetes. In European populations, some of the strongest known genetic risk factors sit in genes like TCF7L2, a variant that substantially raises diabetes risk in people of European descent. When researchers checked the same variant in Chinese and other East Asian cohorts, the association was noticeably weaker. Instead, East Asian diabetes risk turned out to be driven more heavily by a different cluster of genetic variants, some tied to insulin secretion rather than insulin resistance, a distinction that actually matters for how the disease develops and how it should be treated. This is part of why East Asian populations can develop type 2 diabetes at meaningfully lower average body weights than European populations typically do, a pattern doctors noticed clinically for years before the underlying genetics caught up to explain it. Drug metabolism turned out to be an even bigger story, and this is the part of the research with the most immediate real-world consequences. One of the clearest examples involves a gene called CYP2C19, which controls how the liver processes a wide range of common medications, including certain blood thinners and antidepressants. A significant proportion of East Asian populations, in some studies close to a third, carry variants that make them poor metabolizers of drugs processed through this pathway, a rate substantially higher than in European populations. That matters enormously for a drug like clopidogrel, a blood thinner prescribed after heart attacks and strokes to prevent dangerous clotting. In poor metabolizers, the body can't properly convert the drug into its active form, meaning patients can take the medication exactly as prescribed and still not get meaningful protection against a second stroke or heart attack. This isn't a hypothetical concern. It's precisely why regulatory agencies eventually added specific warnings to clopidogrel's labeling, and why hospitals treating East Asian patient populations increasingly screen for CYP2C19 status before relying on the drug at all. Then there's ALDH2, probably the single most famous population-specific gene variant in all of human genetics, even if most people don't know its name. This gene controls how the body breaks down acetaldehyde, a toxic byproduct created when alcohol is metabolized. A specific variant of ALDH2 that essentially disables this enzyme is carried by a substantial percentage of East Asian populations, roughly a third to well over a third depending on the specific population sampled, and is almost entirely absent in European populations. People carrying this variant experience the well-known alcohol flush reaction, rapid facial redness, increased heart rate, nausea, after drinking even small amounts of alcohol because their bodies can't clear the toxic byproduct efficiently. It's not a myth or a stereotype. It's one of the most well-documented single-gene population differences in existence, and it carries real medical weight too, since carriers of this variant, who drink regularly anyway, face a significantly elevated risk of esophageal cancer, because their tissues are repeatedly exposed to a toxic compound their bodies can't properly break down. Chemotherapy drugs showed similar population-specific quirks. 5-fluorouracil, a chemotherapy medication used widely in cancer treatment, showed strikingly different risk profiles for dangerous side effects between East Asian and European cohorts in pharmacogenomic screening studies, driven by differences in the frequency of variants that control how the drug gets broken down in the body. Blood pressure medications, certain anticoagulants, even some pain medications showed measurably different effectiveness and side effect profiles depending on population ancestry. None of this is about one population's biology being better or worse. It's about the basic reality that drugs designed and dosed almost entirely around European genetic data don't always translate cleanly to a population with a meaningfully different distribution of metabolic gene variants. And for a long time, nobody had the data to even notice. This is where the story gets genuinely uncomfortable for the medical establishment, because it means decades of drug development built almost entirely on trial populations of European descent, quietly built in a blind spot. A recent large-scale comparative study integrating data from the 1000 Genomes Project with the pharmacogenomics knowledge base found substantial systematic differences between East Asian and European populations across hundreds of genetic markers, tied specifically to drug response, drug metabolism, dosing requirements, and the likelihood a patient would need to discontinue a medication entirely due to adverse reactions. The researchers weren't hunting for controversy. They were simply trying to quantify how much of modern precision medicine, built on overwhelmingly European genetic data, might be silently miscalibrated for everyone else. The Hong Kong Genome Project added another layer to this picture. Researchers there sequenced the genomes of over 20,000 participants and compared the results against genetic screening panels originally built using European ancestry data. When they cross-checked the panels against the actual Chinese population genomes, they found dozens of clinically important genes that would have been completely overlooked using the European-designed panel alone, genes tied to recessive disease conditions that simply weren't well represented in the original screening tools. In practical terms, that means a genetic test built and validated on European populations could, if applied uncritically to a Chinese patient, miss disease risks that a properly calibrated panel would have caught immediately. Cancer genetics turned out to carry some of the sharpest population-specific signals of all. Nasopharyngeal carcinoma, a rare cancer type in most of the world, occurs at rates dramatically higher in southern Chinese populations, particularly in Guangdong province, than almost anywhere else on Earth, so consistently that doctors have nicknamed it Cantonese cancer for decades. Researchers eventually traced part of that elevated risk to specific variants clustered in the HLA region, a dense stretch of the genome that controls immune recognition, combined with a strong environmental trigger from Epstein-Barr virus exposure and traditional preserved food consumption. It's a case where a genetic predisposition common in one population interacts with a shared environmental exposure to produce a cancer risk profile almost unheard of elsewhere. Liver cancer tells a similar story. Hepatitis B infection rates are far higher across East Asia than in Europe, for historical and epidemiological reasons, and researchers have identified specific genetic variants, more common in Chinese populations, that influence how the immune system responds to chronic hepatitis B infection, and how likely that infection is to eventually progress toward liver cancer. None of this means Chinese populations are simply more prone to cancer, or it means specific cancers cluster around specific gene-environment combinations, and those combinations are not evenly distributed across the human species. The HLA region deserves its own mention here because it turned out to be one of the most dramatically different parts of the genome between Chinese and European populations, and one with enormous practical stakes. HLA genes control how the immune system distinguishes the body's own cells from foreign invaders, which makes them critical for everything from organ transplant matching to vaccine response to autoimmune disease risk. The specific HLA variants common in Chinese populations are, in many cases, barely present in European donor registries at all, which is precisely why patients of Chinese and broader East Asian descent have historically faced significantly longer waits and lower match rates for bone marrow and organ transplants in countries where donor registries skew overwhelmingly European. It's a stark, real-world consequence of exactly the kind of population-specific genetic architecture these studies were built to uncover. And it's part of why transplant registries worldwide have pushed hard in recent years to recruit more donors specifically from underrepresented ancestries. Cardiovascular genetics added yet another layer. A cluster of variants near a stretch of chromosome 9, often referred to by researchers simply as the 9p21 region, is one of the most robust known genetic risk factors for coronary artery disease in European populations. When researchers checked the same region in Chinese cohorts, the variant was present, but its effect size and population frequency didn't line up cleanly with what European studies had predicted, meaning a risk score built entirely on European data would misjudge Chinese patients in either direction depending on which specific variant combination they carried. Cholesterol and lipid metabolism genes showed comparable mismatches, part of why the Cell Genomics study comparing Chinese and European metabolite genetics specifically flagged how much of existing lipid and cardiovascular genetic research had been built almost entirely around UK biobank-style datasets, meaning a huge fraction of the genetic map for something as common as heart disease had simply never been properly checked against the population, making up nearly a fifth of humanity. Even Alzheimer's research, one of the most heavily studied areas in all of human genetics, showed measurable population differences. The APOE gene, and specifically a variant called APOE4, is the single strongest known common genetic risk factor for late-onset Alzheimer's disease. It's well studied in European populations, where carrying one copy meaningfully raises risk, and carrying two copies raises it substantially further. Studies in Chinese and other East Asian cohorts have found that APOE4 carriers still face elevated risk, but the size of that risk increase and the way it interacts with other factors like diet and cardiovascular health doesn't map perfectly onto the numbers derived from European studies, which matters enormously for how doctors counsel patients and families about their own individual risk. If you're finding this as surprising as we did while researching it, go ahead and hit that like button. It genuinely helps this reach more people who are curious about what their own DNA test results actually mean. And there's more, because the story isn't just about disease and medication, it stretches into some of the strangest corners of human deep ancestry too. Beyond disease risk and drug metabolism, researchers found genuinely fascinating population-specific fingerprints buried in adaptation to extreme environments. Tibetan populations, for example, can live and function at altitudes that would cause severe altitude sickness in almost anyone else on Earth. For years, this was one of the best documented cases of ancient interbreeding shaping modern biology. Tibetans carry a variant of a gene called EPAS1, which helps regulate how the body responds to low oxygen levels, and that variant traces back to Denisovans, the archaic human species first identified from a finger bone in a Siberian cave. Somewhere in the deep past, ancestors of modern Tibetans interbred with Denisovan populations, and the specific gene variant that helped Denisovans survive in cold, oxygen-poor environments got passed down and preserved because it offered an enormous survival advantage at extreme elevation. It's one of the clearest known examples of ancient interbreeding directly shaping the biology of a living human population today, and it shows up nowhere near as strongly in European populations, who never needed that particular adaptation. Han Chinese populations at sea level carry a different, unrelated set of variants in oxygen metabolism genes compared to Europeans as well, not offering the extreme altitude tolerance seen in Tibetans, but subtly tuning how efficiently oxygen gets used at the cellular level. It's a reminder that human populations weren't just passively drifting apart genetically after leaving a common ancestral population. They were being actively shaped, gene by gene, by the specific environmental pressures each group happened to face over tens of thousands of years, from thin mountain air to starch-heavy river valley diets to the low gray sunlight of northern Europe. There's also a genuinely strange archaic ancestry angle buried in all of this. East Asian populations carry small but measurable amounts of Denisovan ancestry, generally more than European populations, which carry almost none, reflecting different migration and interbreeding histories after the initial exodus from Africa tens of thousands of years ago. Some East Asian populations, particularly in Southeast Asia, carry Denisovan ancestry from what current research suggests may have been more than one distinct Denisovan population, adding yet another layer to the puzzle of exactly how modern East Asian ancestry took shape. None of this is invisible, undocumented mystery DNA. It's measurable, published, peer-reviewed archaic ancestry, but it does mean that Chinese and European genomes carry meaningfully different inherited fragments from extinct human relatives, on top of everything else that diverged after the two populations split. There's a quieter, less headline-grabbing layer to all of this too, involving the gut microbiome. Gut bacteria composition is shaped by a mix of diet, geography, and host genetics, and several comparative studies have found that Chinese and European populations carry meaningfully different average gut bacterial profiles, partly tracking the same starch versus dairy dietary split reflected in genes like AMY1 and lactase persistence. Bacteria specialized in breaking down complex plant starches and fiber tend to show up more heavily in gut samples from populations with a long history of grain-heavy diets, while bacteria adapted to processing dairy fats and proteins show up more in populations with a long history of pastoral, dairy-heavy diets. It creates a feedback loop that researchers are still working to fully untangle. Genes shape which foods a population can efficiently digest. That dietary pattern shapes which gut bacteria thrive over generations. And that bacterial community in turn influences everything from immune function to metabolic disease risk to how efficiently a person absorbs nutrients from a given meal. It's a reminder that the genetic story here doesn't stop at the DNA itself. It ripples outward into the trillions of microbial cells every person carries too, each shaped by thousands of years of the same evolutionary pressures. All of this adds up to a conclusion that the researchers behind projects like ChinaMap have been fairly careful and consistent about stating clearly. Human populations are not genetically interchangeable in every respect, Different groups experience different evolutionary pressures, different diets, different climates, different disease environments, and different chance encounters with archaic human relatives, and all of that left real, measurable, medically relevant fingerprints in the genome. At the same time, and this is the part that gets lost whenever this topic goes viral online, the genetic differences between any two individuals within the same population are consistently far larger than the average differences between populations as a whole. If you compared two random people from Beijing to each other, their overall genomes would differ from one another, far more than the average Chinese genome differs from the average European genome. What these studies are picking up on isn't some sweeping genetic gap between two categories of humans. It's a difference in the pattern and frequency of specific, well-defined variants, mostly tied to metabolism, drug processing, and environmental adaptation, layered on top of a genome that remains overwhelmingly shared across all of humanity. That distinction matters enormously for how this research actually gets used. The honest, responsible version of this story is not about ranking populations or suggesting one group's biology is somehow superior. It's about closing a dangerous blind spot in modern medicine, one built by decades of genetic research that treated European populations as the default template for the entire species. Pharmaceutical companies are now increasingly required to run separate clinical trials in Asian populations specifically because of findings like these, since a drug's approved dose and sometimes its entire safety profile can look meaningfully different once you account for population-specific metabolism genes. Genetic testing companies are beginning to build ancestry-informed risk models rather than one-size-fits-all scores calibrated almost entirely on European data. Hospitals treating diverse patient populations are increasingly screening for genes like CYP2C19 before prescribing certain heart medications, specifically because ignoring population level pharmacogenomics has already led to real, documented cases of ineffective treatment. The uncomfortable truth underneath all of this is that for a very long time, human genetics quietly meant European genetics, simply because that's whose DNA filled the early databases and clinical trial rosters. Projects like ChinaMap, the Taiwan Biobank, the Hong Kong Genome Project, and comparable initiatives now underway in Japan, Korea, and across Southeast Asia are, piece by piece, correcting that imbalance, and every time they do, they uncover another layer of genuine, clinically meaningful difference that European-centric medicine had simply never been positioned to see. None of it suggests some hidden hierarchy between populations. What it suggests is that human genetic diversity is far richer, far more specific, and far more shaped by deep history than the tidy, one-size-fits-all model of human biology that dominated medical research for most of the 20th century. So, when researchers finally put Chinese and European genomes side by side at real scale, what shocked people wasn't the discovery of some secret hierarchy hiding in the DNA. It was the realization of just how much modern medicine had been quietly built around only one slice of humanity, and how much better, safer, and more accurate healthcare could become once that blind spot finally started closing. Every population on Earth carries its own deep signature of the specific climates, diets, diseases, and even extinct human relatives its ancestors encountered along the way, written directly into genes that still shape how our bodies respond to food, sunlight, altitude, and medicine today. The differences are real, they are measurable, and understanding them properly, rather than ignoring them or misusing them, is turning out to be one of the most important frontiers left in modern medicine. If this changed the way you think about your own DNA, drop a comment with the part that surprised you most and subscribe. Because next week we're covering another population whose genome revealed something modern medicine had completely missed. You will not want to miss it. Thanks for watching, and we'll see you in the next one.